So welcome everyone. I will talk about our research on the differential impact of birth weight and gestational age on newborns' brain responses to language. First of all, to study brain functions in newborns, near-infrared spectroscopy, or NIRS in short, is an exceptionally suitable method offering a non-invasive, safe and silent approach to measure neural activity. By tracking changes in oxygenated and deoxygenated hemoglobin, NIRS provides insights into the hemodynamic responses of the infant brain, which are metabolic correlates of neural activation. This technique is particularly well suited for newborns due to their thinner skulls and minimal hair, allowing the infrared light to penetrate deeper into the brain and thus providing clearer signals from cortical regions. These hemodynamic responses can be influenced by various physiological factors that impact blood flow, overall brain development, and maturation. So understanding these factors is essential for interpreting NIRS data and drawing accurate conclusions about early neural development and functioning. So the current study sets out to investigate two factors that may influence brain hemodynamics in newborns: birth weight and gestational age at birth. When investigating how these perinatal physiological factors influence newborns' brain responses, speech perception is a highly relevant cognitive ability that can be used as a model. So as the auditory system becomes functional around the third trimester, it enables fetuses to perceive auditory stimuli such as the mother's voice and environmental sounds already in the womb. So speech and language are some of the primary sounds a fetus encounters during the intrauterine period, making them especially relevant for studying the effects of gestational age, which basically determines the duration of prenatal exposure. And birth weight, a commonly used indicator of overall prenatal health and the single best predictor of later developmental outcomes, may influence how effectively these early auditory experiences are processed by the brain. Linguistic tasks are particularly sensitive to detect variations in regional brain activations and specializations, such as the left hemisphere dominance for language, which develops early in life and may be disrupted by perinatal factors. Also, linguistic stimulation during pregnancy contributes to the formation of neural connections, which enhances overall cognitive development in newborns. So by examining newborns' responses to speech and language, we can identify how gestational age and birth weight may influence early brain development and brain functions in newborns. Birth weight and gestational age are the most important immediate physiological measures that are easily available at the moment of birth. They are significant predictors of later development and developmental outcomes in general due to their impact on early neural development. Infants born preterm or with low birth weight are at increased risk for developmental impairments, including cognitive, motor, and language deficits, highlighting the importance of the last trimester for critical neurodevelopmental processes. This period is also crucial for brain growth, and low birth weight is associated with reduced volumes of cortical gray and white matter that are essential for cognitive functions. So the additional time for growth and development in utero supports better maturation of the brain and other vital organs, which is reflected in better cognitive performance and fewer developmental delays. And additionally, higher birth weight often correlates with better overall health, reducing medical complications that could interfere with early cognitive development. So these findings highlight the importance of a full-term nurturing prenatal environment indexed by the two most reliable physiological measures in terms of developmental outcomes. The current study therefore aimed to explore the role of prenatal period and examine the effects of perinatal factors on newborns' NIRS responses to speech and language. We investigated how the length of prenatal exposure indexed by gestational age, and overall maturation indexed by birth weight affect newborns' brain activations and linguistic abilities. For this, we used a meta-analytic approach, pulling together data from NIRS studies with newborns and examined the effect of prenatal experience and maturation on early brain development and linguistic abilities. We expected longer gestational periods and higher birth weight to result in stronger brain activations. For this, we analyzed the data of 14 near studies on typically developing newborns' linguistic abilities. The studies examined brain responses to a variety of linguistic tasks like rule learning and prosody detection. The meta-analysis included 192 full-term newborns aged between 1 and 4 days. In the NIRS cap, 8 to 10 sources and 8 detectors were placed on the newborns' heads bilaterally, creating 10 to 12 channels on each hemisphere. Our study focused on the left temporal and frontal brain areas that are crucial for speech perception and language processing, compared to the analogous right hemispheric regions. So importantly, gestational age and birth weight are indices of the length of prenatal experience with language, but also of overall prenatal maturation. It is impossible to fully tease the two apart in our data set, but as our best attempt to distinguish between the maturational effects and the effects of prenatal language experience, we correlated two types of NIRS measures with our perinatal variables. The first type of NIRS measure is the absolute magnitude of the NIRS response, which is the size of the NIRS response in a given experimental condition compared to baseline. As an example, on the figure, the continuous red line is the NIRS response for the control condition, and the dashed pink line is for the experimental condition, which were both compared to baseline. These can reasonably be expected to be more impacted by maturation. By contrast, the differential NIRS response, which is the ability to discriminate between various experimental conditions, in this case, the difference between the pink and the red lines, may be more specifically linguistic. So if gestational age or birth weight correlates with the magnitude of the NIRS response, then it may be interpreted as a prenatal maturational effect, while if correlations are found between gestational age or birth weight and the differential NIRS response, then it's more likely that it's not simply maturation, but also the length of prenatal experience that also plays a role. We used linear mixed effects models to analyze brain activations and effect sizes in these three comparisons. In the meta-analytic model, the random effect structure included random intercepts for subject, study, and lab to account for variability at the different levels. In case of failure to converge, the structure was gradually simplified by removing the random intercept for lab first, followed by study, and then subject. Fixed effects included birth weight, gestational age, region of interest, and hemisphere. These variables were added one by one to a model, and the resulting models were compared. The model with the lowest AIC value was selected as the best fitting model, and the factors in this best fitting model were used to analyze their differential impact on brain activation. These analyses were conducted separately for oxygenated hemoglobin and for deoxygenated hemoglobin. In the examination of oxygenated hemoglobin for the control versus baseline comparison, there was a marginal interaction between birth weight and hemisphere, showing stronger activations in the right hemisphere. This effect was found to be driven by the frontal regions. So this result suggests that higher birth weight is associated with a better overall brain development, showing more robust neural activity, especially in brain areas that are involved in higher order cognitive functions like language, such as the frontal regions. But there were no significant effects for the magnitude of the NIRS response to the experimental condition or for the differential NIRS response. In the case of deoxygenated hemoglobin, let me first remind you that deoxygenated hemoglobin decreases with activation, so more negative responses mean stronger activations. So for the control versus baseline comparison, there was a significant interaction between region of interest and hemisphere, showing stronger activations in the temporal regions of both hemispheres, which is illustrated on the bar graph on the left. And there was another significant interaction among birth weight, region of interest, and hemisphere, and it showed that while birth weight modified the relative contribution of the temporal and the frontal regions in the left hemisphere, it didn't in the right hemisphere. Specifically, increasing birth weight led to stronger activations in the temporal regions of the left hemisphere, including the primary auditory cortex. And there were no significant effects for the magnitude of the NIRS response to the experimental condition in this case either, but for the experimental versus control condition, Significant effects were found for region of interest and for its interaction with birth weight and with gestational age. In the interactions, birth weight was associated with stronger activations in the frontal regions, which you can see on the upper right graph, while gestational age was associated with stronger activations in the temporal regions, shown on the lower right graph. This suggests that gestational age may affect auditory discrimination in the temporal regions, where the auditory cortex is located, while birth weight may represent the maturation of frontal regions, including Broca's area, which are involved in higher order language tasks. The models mostly showed a significant effect of birth weight and less so for gestational age, suggesting that biological growth indexed by birth weight might be more important than the length of prenatal experience. And surprisingly, the results were stronger for deoxygenated hemoglobin compared to oxygenated hemoglobin. While oxygenated hemoglobin is more sensitive to cognitive manipulations in typical experiments with newborns, our results suggest that deoxygenated hemoglobin may be more sensitive to overall biological maturation, showing a physiologically more robust response. But our study has some limitations. Firstly, the different types of linguistic stimuli used by the studies that we included in our meta-analysis could affect the comparability of the results. And secondly, the samples included naturally conceived newborns, so gestational age is just an estimate that may lack precision, and this imprecision could affect the exact developmental stage and linguistic abilities of the newborns. Future research could explore how gestational age and birth weight affect neural pathways over time with longitudinal studies, and also expanding the research to preterm and low birth weight infants could further enhance our understanding. Overall, the current study highlights the significant influence of biological maturation indexed by birth weight on early brain development and language acquisition. And it also reveals a stronger correlation of deoxygenated hemoglobin with prenatal experience and maturation, arguing for the need to use deoxygenated hemoglobin alongside oxyhemoglobin in newborn ear studies. And thank you.
Thank you, Noémi. We have some time for a few questions. Is there a question there? No. Before Luca, I just have a random question. So, do newborns who are born before the 9 months, does the time they spend in the incubators compensate for potential loss that they might have had?
No, so preterm infants spend more time in hospitals where they hear the everyday hospital noise instead of speech or their mother's voice that they would hear in the womb. So that's the idea, that if they would hear speech or their mother's voice also in the incubator, then it would compensate for the developmental delays, or it would help their brain maturation.
Thank you. We'll have Luca.
Sorry, this is a clarification question because I was not sure I understood well one thing. You didn't find any effect of the language conditions?
Yes, so also the experiment...
So my question was, is it... So first, if I understood well. The second is, were you expecting any effect? I mean, I'm not very sure to what extent this was something that you are neutral with respect to, or whether you had an investment into these results.
Mm-hmm. So both the experimental condition and the control condition were including linguistic stimuli, and we found significant effects for the control condition. So why we didn't find any significant effect for the experimental condition is maybe because that we included 14 studies in which some of them they found significant differences, in others they didn't, so maybe the overall brain activations that...
So it's variability of the kind of
yes.
designs. Okay,
yes, yes.
Thank you.
We still have some time if there is any more questions. Okay, thank you, Naomi. With this, we conclude the session on language.